of the big areas of proteins, and one that we're really going to focus on for the next couple screencasts, are enzymes. Enzymes are typically large globular proteins. And these are present in every cell of the body. They act as catalyst. Right? And I want you to remember catalysts are just compounds that accelerate reactions of metabolism, but aren't consumed or changed by those reactions. And we've talked about catalysts before when we talked about rates of reactions. An enzyme can't force a reaction to occur that would not normally occur. An enzyme simply makes the reaction faster. The enzyme name normally is going to appear above or below the arrow, and most enzymes end with the ASE ending there. Now, when we talk about enzymes, you're going to hear the word active site. And the active site, that's just the part of the enzyme where the reaction occurs. And the reactants here are called substrates. We also have some other players in these reactions too. One of those players are things called cofactors. These are inorganic substances, like metals, for example. Okay. And then we also have coenzymes. And coenzymes are just small organic molecules that help with biological reactions. When we look at this, for example, glucose is going to react with the molecule ATP. ATP is an energy molecule. And when that happens, this reaction is really mediated by the enzyme hexokinase. And so when we look at the enzyme shown here, it's really large. And we can see glucose is really small, and glucose fits into the active site. Now, glucose is held there by the amino acid side chains and the interactions between those. And then once the reaction occurs, then glucose 6-phosphate and ADP are the products, and they'll leave the active site. And so the enzymes really help facilitate all sorts of reactions in our body. I find them to be pretty amazing. Now, there are a lot of different models for how enzymes work. One of the things that you're going to hear, one of the vocabulary words that we use is an enzyme substrate complex. And what we see here is that is just the complex when the enzyme or the substrate is in the enzyme, for lack of a better term there, right? And so when the substrate or the reactant binds to the enzyme, that's called the enzyme substrate complex, and that's abbreviated with an ES. Now, there's a lot of different models to describe how the enzyme substrate complex forms. One is a lock and key model, and basically with this model, the active site is rigid. It's a rigid, inflexible shape that is an exact complement to the substrate. Very similar to when you put a key in a lock. And there's an example here, right? We've got this shape, it'll fit in this enzyme. Here's the enzyme substrate complex. That's lock and key. We can also have an induced fit model. And I want you to notice an induced fit model, there's a rigid shape here, but this shape doesn't exactly match that one. And with the induced fit model, the idea there is that the enzyme's active site is flexible and roughly complementary to the substrate. And so basically the enzyme's active site is adaptive and it'll adapt to that. This is an example of the glucose and hexokinase interaction through induced fit. Notice that, so here's glucose, here's the enzyme, right? And notice that when glucose enters the active site of hexokinase, the enzyme's right and left side, and these are called lobes, they come together and they wrap around glucose snugly, kind of like giving glucose a little hug there. And that's really where that kind of conformation changes. And so that's an example of induced fit. Now, catalysts speed up reactions by lowering the activation energy. We've talked about this before. Stop. I have a cat that's trying to steal my pretzels. We have talked about this before. So, catalyst speeds up a reaction by lowering the activation energy right? And this is accomplished during the formation of the enzyme substrate through interactions between the enzyme and the substrate. When that enzyme substrate forms, the substrates are in really close proximity and they don't have to find each other as they would as if they were in solution. And so you can see here, if we had an uncatalyzed reaction, this would be the activation energy, and you need a lot of energy for the reaction to happen. But with an enzyme, you don't need as much energy for the reaction to happen. So proximity is very important as for our enzyme reactions. The other thing that enzymes help with is orientation. So in the active site of an enzyme, the substrate molecules are held at the appropriate distance and in correct alignment to each other to allow the reaction to occur. So the substrate molecules are held at the appropriate distance and in correct alignment to each other to allow the reaction to occur. One of the things that helps with this is the arrangement of the amino acid side chains that creates interactions that orient the substrate so they'll react. Without an enzyme to orient the substrates bumping into each other in solution may not react because of incorrect positioning. So we see this here, right? In the absence of the enzyme, it's more difficult for the substrates to get close enough to orient themselves to react appropriately. But if there's an enzyme there, the enzyme says, okay, look, you green arrow thing, you're going to go here. And then the yellow ball thing is going to go here, and it's going to be easier for the substrate to get close enough and orient themselves in the reaction. And the correct orientation lowers the activation energy. So that's another thing that enzymes do to lower the activation energy. And then the third thing, so we've got proximity, we have orientation, and then the third thing is bond energy. And when an enzyme interacts with its substrate to form the enzyme substrate complex, the bonds of the substrate molecule are weakened. Right? And those weakened bonds means that they will more readily react. In other words, weaker bonds break more easily and the activation energy is lowered by this effect. And so you can see this here, right? If this is the enzyme active site, right? You can kind of imagine the substrate here is held in, and then the bonds are weakened, allowing this to react more easily and lowers the activation energy. In the next screencast, we're going to look at other factors that affect enzyme activity.